Hi everyone. I'm presenting a paper that was actually published in October in Nature. It was also actually featured as on the cover of the Nature journal. And this paper was about nanobody-based recombinant antivenom. So let's get into the paper. So what this paper basically discussed was that they have made a new antivenom for treating snake bites for some species in Africa. But why is snake bite even a problem? So let's first discuss what is the snakebite crisis in the world. So snakebite has been actually recognized as a neglected tropical disease by WHO, and they also have been making strategies to reduce or cut down the deaths that have been caused by snake bites by 50% by 2030. So, as part of this mission, a lot of different laboratories in India as well as other parts of the world are working to develop better therapeutic strategies because if we look at it, snakebite is actually disease of the poor, and predominantly I have also like in this map you can see that the snakebite related deaths are widely present in India, in sub-Saharan Africa, and also in Latin America. So it's not like spread everywhere. It's mostly near in the tropical regions. And currently, the strategy or the therapeutic strategy that is used for treatment of snake bites is a normal antivenom which is derived from horses. So now, if anyone who has a immunology or anyone has a molecular biology background can understand that these antivenoms are derived from other animals. Therefore, whenever we get these antivenoms as therapeutics, these might lead to side reactions because these horse-derived proteins are actually recognized as foreign by our immune system. So, therefore, there have been a lot of problems with the current antivenom strategies because this is a 100-year-old technology. And not only this, but these are serums, right? And all these serums need cold chain or refrigerated supply because and this adds to the cost. Not only this, when I will proceed in this like my presentation, I will also tell you that the venom is a very complex cocktail and only some of these toxins are clinically relevant. Due to which what happens is that whenever these when you immunize horses with this whole venom, the amount of antibodies against that clinically relevant toxin is very less. Therefore, you need multiple vials for the treatment. And as I emphasize that this disease is disease of a poor. Therefore, not all these poor people after bitten by a snake can afford these expensive treatments. So yeah, these are the current problems that are there with the current therapeutic strategy. And as I said, that everybody has been trying to generate better therapeutic strategies. Now, there are a lot of different strategies that can be possible, but you can understand that this not everyone will be willing to get vaccinated for this disease because you are basically getting vaccinated for a toxin, and that toxin, even after being inactivated, might work. So, therefore, the only strategy that is possible is that you give a therapeutic treatment. You cannot get a preventive treatment in this case, you need a therapy. So, the treatments that are currently being produced for these are either drugs that can be given after you have been bitten by a snake, or there can be these antibody cocktails or nanobody cocktails. So first let's look at what is the composition of the venom and what venom these people or what snake species this group has targeted in their paper. They have also discussed that yeah, around 3 lakhs to 4 lakhs envenomings happen annually, and out of these, around 7,000 30,000 deaths happen in Africa because of these species, these families of snake, that is cobras, mambas, and wrinkles. And not only this, the other problem is that even if you're bitten by a snake, even if you don't die, your organs might or your limbs might get affected, and you might it might lead to amputations. And once these amputations happen, you are basically losing your limb. And since I said that all these snakebite cases are mostly common in poor countries like India or sub-Saharan Africa, therefore a person, due to this amputation, a family might lose a sole breadwinner of their family. Therefore, it is very important to prevent this neglected disease. So, as I was discussing, that venom is not just one protein, it's a cocktail of multiple proteins. Not only just proteins, but also some non-enzymatic proteins and some other components like DNAs, RNAs as well. So yeah, it's a complex cocktail, and and a lot of studies have happened where they have recognized what are some clinically relevant toxins that need to be targeted in particular species. So if we look at it, so in these three species, that is cobras, mambas, and wrinkles, the three major toxins that need to be targeted are neurotoxins, cytotoxins, and PLA2s. So I will briefly explain how all of these toxins works. If we look at neurotoxins, as the name is self-descriptive, these basically target your neurological machinery, and these basically bind to your NACHRs, these which are the nicotinic acetylcholine receptors. So now once the neurotoxins bind to this, they prevent your neural signal transmission and cause paralysis. Now, if you look at this, if you like, there's paralysis in your hand, you can work with it. But what if your heart muscles or your lung muscles get paralyzed? In that case, there's a complete respiratory failure, and the person who is bitten by snake that has neurotoxins dies. That is how neurotoxins cause death. Then we have cytotoxins. Now, cytotoxins are basically a subfamily of neurotoxins, and what they do is cyto, which means cells, and toxins. They target your cells. They target your cell membranes, and they cause tissue death. And because of this, what happens is that your tissue gets necrotic, and you have to basically lose your limb or your finger. So this is what is leading to the amputations. And then we have PLA2s. Now again, phospholipase. It's a class of phospholipase enzymes that we have in our body, but it's a different isoform. And what these does is they basically target phospholipids, which are again present in your cell membrane, and these lead to inflammation, pain, and like the whole necrosis. Yeah. So these are the figures also by which you can like understand, but I've like basically explained through verbal explanation. So now, as I was discussing, there are various methods to deal with snakebite now. Like people have been trying to provide different innovative solutions. But one solution that these people have been discussing are nanobodies. So first we need to understand what is a nanobody and how it is different from a conventional immunoglobulin, like an antibody, a human antibody. So we have in our body we have normal immunoglobulins, and one of those immunoglobulin classes IgGs, and this is the structure of immunoglobulin, where we have this FC region, and this is the antigen binding region, and this is generally constant. It is called FC or constant region. It has these CH domains, and then this extends up to the Fab, that is the antigen binding fragment as well. But one special thing in our IgG antibodies is that they have a heavy chain, which is this blue ones, and we have a light chain. And this CDR region is the one that binds to the antigen and provides specificity to the antibody. Now, when you break these, so you get this, if you break this region, you find these Fab fragments. Or what you can do is that you can break this also, and you can just have these variable regions, that is the variable heavy and the variable light region, and this can serve as an SCF, your single chain fragment variable. Now what happens is that in case of camels, in case of llamas, they have certain special antibodies that lack these light chains, which makes it very easy to engineer, and these also become very smaller in size, and hence they can easily penetrate tissues. However, one drawback of this is that they can also be cleared by our body very quickly, but that can be solved by conjugating it with some other thing. So this is what people have been targeting for a lot of diseases. Like they engineer these nanobody, or they target these nanobody regions and engineer these antibodies for their specific purpose, or like a specific disease or specific antigen. So this region is called a VHH, or a single domain antibody or a nanobody. And you can see in the range of size that it's a very small protein. And these are only in certain special organisms like camels and llamas. So how these antibodies are produced, you basically, for any disease, you basically immunize these camels or these animals at sublethal or like sub infection doses. And after you get those blood samples, you basically try to find those antibodies which are specific to your target. So these are some characteristics, as I said, that yeah, it's a single chain antibody, and they can be derived from camels, llamas, and alpacas, and they have high affinity also. They can be easily engineered. And in fact, as I was saying, that since these are much smaller, they are much stable, and because of this high stability, high thermostability, they can be also supplied from one region to another without requiring any cold refrigeration, which cuts down the cost of storage and supply. And these can also be easily expressed because as we can see that these two chains, the heavy chain and the light chain, are actually held by disulfide bonds. But in this, there are no disulfide bond linkages. Therefore, it is very easy to express them in even bacterial systems or prokaryotic systems, which makes it even less expensive to produce. Um, so the core methodology that these people have used in discovering these antibodies is a phage display technology. Now, this is phage display is actually a Nobel Prize winning technology in 20. It won the Nobel Prize in 2018. And so, what basically this technology does is that we know bacteriophages and we know bacteria. Now, bacteriophages are like viruses that can infect bacteria, and they infect bacteria and inject their genetic material, oh, sorry, they acquire the genetic material of bacteria, and hence that is how they expand and like expand their library. So what you do is that if once you have immunized a particular animal, let's say camel, with your toxin, you basically take its blood out, you will isolate B cells from it. And once you isolate these B cells, you can basically take out their DNA, like the total DNA pool from those B cells. Once you have that, you will transform this in bacteria. Now, each of these bacteria will take one plasmid, and this creates a bacterial library. And then what you do, you infect this bacterial library with a bacteriophage. Now what this will now bacteriophage will infect the bacteria, and you will get a bacteriophage library. Now each of these bacteriophage library will have each of these individual plasmids which code for basically the antibody that those B cells were supposed to produce. Can I get a note on if everybody is like on page with this, like if everybody understood? Otherwise I can explain this again, but this is the core of this paper. Am I audible?
Yes, ma'am.
Yes, you are.
So like can I get a hands up or some voices, like if this is clear, like till this, like how phage display works?
Yeah, they are writing in the chat box.
Can you just tell me, because I cannot really share my screen. I cannot switch
Uh,
my screen.
Baboya Raseli and Iftikhar and Kirti, they have
No.
got
I
it.
just want a normal, just a overall view that if everybody is able to understand what I'm saying,
Yeah, they are
particular
able
to.
to.
Okay, sure. So I'll go ahead because I just didn't want it to be complicated for everyone. Yeah. So um, each of these phages now basically have plasmids, and each of these plasmids have the capability to express the antibody that originally those B cells were supposed to express. Okay. So now what you will do is that you will take these phages and you will treat them with the antigen. Now only those phages which have an antibody that has the capacity to bind to that antigen, that has the specificity or that affinity to bind to that antigen, will come to your antigen and bind to it. And then you can do multiple washing steps and all of that. And you will basically capture only those bacteriophages that have the antibody specific to your antigen. This is called biopanning. As you can see in this, that you have this displayed protein on the surface of your phages, and this is your target. And now you have basically captured only those phages that have specific antibodies binding to your antigen. And now what you can do is you can just take these plasmids, purify their you can basically take these phages, you can purify their plasmids, and you can express these antibodies in your facility. And then you can proceed to test them. And that is what they have done in this paper. So first they have total targeted 18 snake species which are prominent in Africa. 13 were from cobra, 4 were from the mambas, and one was a wrinkle species. And you have immunized llamas. These llamas will produce specialized these nanobody sorry specialized single chain antibodies. And what you'll do is you'll extract the VHH genes and you will insert them into bacteriophages, as I said. And then you will do biopanning, and then you will select only those phages that have expressed antibodies sticking to your toxins. And then you will isolate these sequences, and then you can just grow them in your bacterial species or any expression system. This is what they have done in the paper. And now all of these sequences that they got, they have screened them to see which of these VHS sequences is better to take it further for the therapeutic or like testing in mice. So yeah, the toxins that we were discussing are these CTX, which are cytotoxins, then small neurotoxins, long neurotoxins, and they've also discussed this one toxin called AGTX. It is present only in a few species. So these are the antibodies that they got. So in total, they got 3,000 VHH clones, okay, that were binding to their toxin. And now from 3,000, they have to go to those specific VHH to see that which are the ones which are most effectively binding, like have the maximum specificity, maximum effect, like affinity, and also bind to only the catalytic pockets of your toxins. Because see, a protein can bind to any part of your body, but it should be able to inhibit your toxin by binding to it. And that can happen only when it's binding to either a catalytic pocket, or it is like sterically causing any modifications. So yeah, so first they have actually they analyzed the binding of each of these antibodies using something known as Delphia. Now, I think all of you must know ELISA. So Delphia is actually a advanced modified version of ELISA, which basically is fluorescence based assay. And instead of using all these enzymes that we use in ELISA, HRP, and all of that, they use some lanthanide based probes, and that's how you get the signal. So now in ELISA, what happens is that if you are doing at different dilutions, so if you're using only one part of your toxin, if you're binding one part of your toxin with one part of your antibody, you might get subsignal. And then if you're taking two parts of toxin, one part of your antibody, like this, you are diluting. So you will get a signal, or you'll get this response curve. So now better is it like if even if at a very low dilution, if your antibody is binding to your antigen and you get a strong signal, that antibody is a better antibody. Right, so you select that. So that is how they have done all these binding assays. If anyone has any question in this later, I will answer. Um, so yeah, that's what they have done using Delphia. It's like, as I said, it's type of ELISA, and that is how they've analyzed each of the response curves to see which of the VHH are actually binding with high affinity to their antigen. So this is how Delphia basically works. So no, sorry. Um, this is how patch clamp works. So what they have actually done is, now once you have selected a particular set of antibodies that yeah, these bind to your toxin, you have to see that is it able to inhibit the effect of your toxin. So there are two things. So first is a neurotoxin. and another one were the cytotoxins, right? So if you are looking at the activity of neurotoxins, neurotoxins have the ability to bind to a receptor, right? Now, if it is able to bind to a receptor, and if you're using an antibody that binds your toxin, it must be able to no, it must be able to inhibit that binding. So if you have an antigen and if you have an antibody, if your antibody is binding to your antigen, now your antigen must not be able to bind to its target, like the receptor. Therefore, in patch clamp, what they do is that if the your toxin is binding to the receptor, you'll get a signal, and if it is being inhibited, then you'll not get a signal. So that is how they have first tested which of these antibodies are able to inhibit the effect of neurotoxins. So they have done wholesale patch clamp, and they have used muscle type NaCHR because that is what most neurotoxins bind to, and they have checked which of these antibodies actually neutralize the short neurotoxins and long neurotoxins. So they have tested a range of these antibodies, and they've also kept toxin only and buffer only controls. And you can see that in buffer only, you see a full signal, a one signal, like it's normalized. And in toxin only, you can see that yeah, the signal has gone very low. But when you are taking an antibody, each of these antibodies is basic at some concentration, are able to bring this back, bring this signal back. Means they're basically inhibiting the binding of your toxin to your receptor. And then now the other class of toxins that they're targeting are cytotoxins and TLA2s, which will basically kill your cell. So they have done something known as a cell viability assay. Now, this is very similar to any cell viability assay like MTT assay and all of those. They have used something known as a WST assay. So it is the same thing that once your cell is live or it is viable, it what it will do is its mitochondria will be active, and because of that there will be a redox potential. And once that redox potential is there, it will cause your substrate to get um, reduced or oxidized, and it will lead to change in color. So more is a change in color means more more percentage of cells are viable. And if there is a less viability, in that case what will happen is that your color change will be less. It will, for example, if your initially your reagent is orange and it turns to yellow after getting oxidized, then no, sorry, after getting reduced, then in that case, this there is more cell viability in your plate. And now, if your toxin is there, what do you expect? You expect the cell viability to go down. Means your orange color will stay orange. It will be less yellow. And if you're using an antibody against your toxin, in that case, what you expect is that there will be more number of cells which will go to yellow. Like there will be more yellow color in your plate. So that is what they have done. So they have all these controls, and they have used whole venoms of all these snakes. So you can see that um, if you have not given if you any antibody, you've given just the venom, the cell viability in this case is almost zero. In Naja nigricollis, there is the um, Nigerian cobra. In this case, you can see that the viability is almost zero, and in the control, it is 100%. And now, if you're giving the antibody of different colors, like for example, this pink antibody, you can see that it is able to help it achieve almost 90% viability. In case of this, it is helping in achieving almost 60% viability. So this is how they are testing the efficacy of each of these antibodies against cytotoxic effects of the antigen. And now they realized that you cannot basically prevent all of these toxins using one antibody. You have to use a cocktail. So if you have 20 antibodies, you have to shortlist what antibodies or what antibody combination works the best. So they have tested knocking out different toxins in different snakes and identified which cocktail will work the best against all these species, 18 species, because you don't want to use all these 20 antibodies because that will also add up to the cost of production. You want to, if you can shortlist just four that can work against all 18, then that is the best strategy, the most inexpensive and economical strategy. So what they did a hit and trial. So if you are so what they did this using in vivo experiments. So in vivo experiments, what you basically do is you take venom and you mix it with the antibody for 30 minutes, and then it's a WHO recommended protocol, because immediately if you are like putting venom and then you are putting the antibody, we also do that. But the first step to do is a pre-incubation study where you incubate your venom with toxin. Whatever interaction will happen, and then you inject that mixture into your mice or your model organism. And if your mice survives, means it survives, and if it dies, means that your cocktail, like your antibody, is not able to neutralize your venom in those 30 minutes. So that's what you do. So in first mice, you have injected just the short neurotoxin, for example, and you can see there's 100% like loss of viability, and survival percentage is zero. All mice die almost instantly, like within the first hour. And then when you use this antibody in 1:1 ratio, you can see that all the mice are surviving. And then you use this antibody, you can see that after a few hours, it is like after a few hours you see so this experiment is actually done over 24 hours. You have to see over like behavior of your mice over 24 hours, and after that you terminate the experiment, you euthanize those mice, and you basically just like sacrifice those mice. So yeah, in this antibody, you can see that after 24 hours, there's only 25% survivability, means like in five mice, only, sorry, in four mice, only one survived. And then in this one as well, this antibody worked in and helped rescuing all the four mice. So you can see now that this antibody also works, this antibody also works. So you can have any one of this antibody in your cocktail, but you will see that which of these two antibodies is able to work against more number of snakes. That antibody will be selected, because as I said, you want to make it more economical, so you have to select only and one antibody that can provide broad effectivity. So then they did this with long chain neurotoxins, and then they tested whole venoms of different snakes, Naja haje, Naja melaleuca, then mamba species also. So they tested all of these in combinations as well. And so, for example, in NHAJ, there are both long neurotoxins and short neurotoxins. So they tested this like both of these, and you can see that yeah, there is 100% survivability when they use this VHH5 AS and TX and VHH9 AL and TX. So this combination you finalize. Now they tested this against Naja and Luca. This worked. Then when they went to this Dendroaspis viridis, now this is a different space, like snake. In fact, this is an entirely different family of snakes. So in this, what happens is that there's an additional toxin, the AGTX and OG toxin. So these different toxins also need certain antibodies. So they added that and they tested all these combinations and cocktails. And based on this, finalized a particular cocktail which contained, I think, around eight antibodies. And yeah, this is what they finally achieved. That first they did pre-incubation experiment to find which of this combination works, and then they did a real case rescue scenario. So in this, what you do is you first, so in life it will not happen, like in real life it will not happen like you get bitten by a snake which is already mixed with antibody, right? In real case, what will happen is that you're bitten by a snake, and you take some time to reach to your primary healthcare center or hospital, and once you reach there, then they will give you a strategy. So this is a real case scenario, and if you want to replicate this in mouse, you have to do a rescue experiment. Now since mouse body is very small, so the circulation in mice will be very quick as compared to our body. So if you're bitten in your hand, it will take some time to reach your leg, but in mice, because of its small body, it will get very fast. It will reach your heart very fast. They might die very quickly. Therefore, there a 5 minute buffer time is given, where what you do is that you inject the venom, then you give it 5 minutes, and then you inject your antibody, and then you see that if your antibody is now able to help your mouse survive or not. So this is what they did. They first gave venom plus PBS, which is no treatment, and you can see they have tested it against a range of snake species, and initially in pre-incubation experiment, like all of these antibodies are working, and when they do the same experiment in rescue setting also, all of these antibodies, the recombinant AVs or the recombinant antivenom, it is working. Now, not only did they do this, they also tested the already existing recombinant and sorry, already existing traditional antivenom that they have, that is Inoserp AV, which is a commonly used um, antivenom in Africa, and they can, you can see that this mixture, rescue mixture, is not able to help the mouse in certain examples. For example, in this case, you can see in Dendroaspis, it is not in Dendroaspis viridis, it is not able to help the animal survive over the course of 24 hours. Even in case of this H. hemacatus, and also in case of N. Hage. So this is what they have shown, that their recombinant antibody mixture is better as compared to the current antivenom strategy as well. Now, the another problem that I discussed was the problem of amputations. That whenever you're bitten by a snake, all these cytoxins also lead to a lot of necrosis on the tissue, and you basically lose your limb because of this necrosis. So they tested that if these antibodies can also help in these help in helping the bitten person by these necrotic effects or these morbid effects. So they tested only three species which are known to have all these dermonecrotic effects. That is this wrinkle species. Then they also tested the two Afronajas, and you can see that when you gave only the venom, there is this necrotic tissue wherever you inject it. So wherever you inject your venom, you can see there is this necrosis. But then when you gave 3-VHH, that is which were again cytotoxins, in pre-incubation setting, you can see that yeah, the dermonecrosis is almost gone. And then when you um, did a rescue experiment where you gave your venom, um, and then after that you just after 5 minutes you gave your antibody mixture intradermally, you can see that it is the dermal necrosis is much lower, significantly much lower in comparison to the venom only controls. And then they also did an IV experiment where what you do is that you give the venom intravenously, and you also give your antibody mixture intravenously. Instead of directly on the tissue, you give it in the blood, and you see if your antibodies are able to reach at the location of your bite and help in dealing with these morbid effects. So you can see in this also that it was able to save from necrotic effects, not 100%, but still like still better. And when you checked it against you checked against it using this Inoserp AV, that is the antivenom that currently is there, you can see that it is also not able to help the individual save from dermonecrosis. So in all, to conclude, you can see that the antibody cocktail that they have is helping in surviving from all these snake species except green mambas, because they so out of 18, 7 they were these this cocktail was able to save them from 17 species, and only green mamba did not survive because of unique dendrotoxins that they might have, against which you can generate another antibody, or maybe they could have supplied it with some other neurotoxin targeting drug. But yeah, this study basically is a no, so basically you're not using any animal proteins, you are making it in factories, so there will be no serum sickness, no anaphylactic shock. So this strategy is definitely safer, and again, as I said, it reduces the cost of storage, it also reduces the cost of like using animals, maintaining animals, and then it is also thermostable, and it solves the problem of cold chain supplies. So yeah, this is it about the presentation. Regarding what I work on, so I am trying to replicate similar strategies, but to target Indian snake species. So in India, so to provide a context, India is actually the snakebite capital of the world, and most snakebite-related deaths and amputations happen in India. And in India, most snake bites are caused by four snakes, which are called the big four snakes of India, which is the Russell's viper, the saw-scaled viper, and the spectacled cobra, and the common krait. Only these four species cause most of the deaths. There are also other neglected but clinically relevant species in different regions, like Western Ghats or northeastern states, or some vipers in the Himalayan regions. And people are first trying to solve the problem of big four, and there's also a focus on solving problem for other clinical relevant snakes. So my target species are kraits, and the we have actually different toxins. So I am targeting PLA2s and neurotoxins, and we are trying to develop similar antibody strategies, but using yeast display, which works similar to phage display. And we have been currently, as our group has published a lot of papers on developing other strategies as well. They have used they have repurposed other FDA approved drugs as well, which has worked great across the country for Russell's viper bites, which actually causes 50% of the bites. And yeah, that's the presentation. That's my time. I'm ready for any questions that you might have. Thank you.